Hello everyone. It is my great honor today to share our recently published work in the journal iMeta, titled, Antiviral Defense Systems Drive Persistence of Antimicrobial-Resistant Bacteria but Limit the Transfer of Antimicrobial Resistance Genes in Anaerobic Digestion. Our research is grounded in the severe current One health crisis, antimicrobial resistance, or AMR. As the data shows, nearly 4.7 million deaths globally were associated with AMR in 2021, while approximately 73% of global antibiotics are used in livestock farming. This massive amount of veterinary antibiotics makes livestock manure systems one of the most critical environmental reservoirs for AMR. Since anaerobic digestion is a primary method for treating livestock manure, conducting in-depth research on this typical ecological interface is highly representative and crucial for addressing the broader AMR crisis. In anaerobic digestion, phage-ARG interactions present two core paradoxes. First, the phage paradox on the left. Phages can lyse and clear ARG, but also spread ARGs through transduction. Reduction versus dissemination, which dominates. Second, the ADS paradox on the right. To survive, ARG arm themselves with defense systems. While ADS protects ARG from clearance, it may also block incoming plasmids, inadvertently suppressing horizontal gene transfer. This remains to be verified. To answer these questions, we operated a 440-day anaerobic digester across 6 operational stages. Multi-omics revealed that thermophilic conditions significantly enhanced ARG reduction, while increased solids enriched them. Interestingly, we observed a silent majority. Around 70% ARGs were plasmid-borne, yet most remained silent. While Bacillota hosted the most ARGs overall, the actively expressed ones were paradoxically concentrated in Pseudomonadota. Meanwhile, viral profiling showed that 69.5% of them were lytic phages. By integrating historical CRISPR records with ongoing high-C infections, we built a comprehensive interaction network. We found that ARG-carrying phages are exceedingly rare, just 0.55%. While we detected 45 transduction events, they are not the dominant force. Instead, phages driving ARG reduction significantly outnumber those driving transduction. Crucially, thermophilic conditions weakened transduction but remarkably amplified this reduction effect. This confirms our core finding: in this system, lysis overwhelmingly outweighs transduction. Faced with intense lytic pressure, ARG were forced into a comprehensive arms race. As shown, ARG carry and express significantly more defense systems, including CRISPR-Cas spacers, than normal bacteria. This provides powerful protection, allowing ARG to persist. However, CRISPR also attacks exogenous sequences carrying ARGs, effectively blocking plasmid-mediated horizontal gene transfer. This illustrates the dual role of these antiviral defense systems. To experimentally validate these omics findings, we conducted further in vitro tests. Using lytic phages, we successfully induced phage immunity in both the donor and recipient strains, as shown on the left. We then investigated how this newly acquired immunity affected the frequency of plasmid conjugation. The results were striking. As shown in the bottom right chart, the emergence of phage immunity significantly reduced the conjugation frequency of the multi-drug-resistant RP4 plasmid between the two strains. Specifically, the transfer frequency dropped by over 98.7%. This provides solid phenotypic evidence confirming the inhibitory effect of phage immunity on the horizontal transfer of ARGs. To summarize the main conclusions of this work, first, by integrating next and third-generation multi-omics, high-C technology, and pure culture experiments, we successfully transformed the invisible phage bacteria war in microenvironments into a clearly visible and verifiable interaction network. Second, we demonstrated the role of phages in suppressing antimicrobial-resistant bacteria. Their reduction effect vastly outweighs the risk of transduction transmission. Finally, we clarified the double-edged sword effect of antiviral defense systems. Although resistant bacteria survive thanks to these defense mechanisms, they pay a heavy price by losing the ability to acquire new resistance genes. That concludes the main content of our research. Thank you all very much for your time and attention.